Hi, everyone. So this is our CRISPR application project on CRISPR in HIV and AIDS. So first, the introduction. What is CRISPR? So CRISPR stands for Clustered Regularly Interspaced Short Palindromic Repeats. CRISPR is a revolutionary gene editing DNA technology. CRISPR comes from the immune system of bacteria and archaea, also known as prokaryotes. CRISPR has been modified to cut specific sequences of DNA to allow for the insertion or deletion of desired genes. This has allowed scientists to experiment with gene therapy, treating diseases and cancer, and genotyping of bacterial strains.
CRISPR-Cas system employs targeting techniques that could minimize viral escape by HIV-Rev encoding gene and superinfection. The goals of CRISPR are to remove HIV genome from the normal human genome, to eradicate the reservoirs of HIV virus, and to inherit the trait to offspring so they would be resistant to HIV. The CRISPR-Cas techniques present a solution through gene therapy by targeting both the provirus and host genes of the virus. The application of CRISPR-Cas editing targets integrated viral genomes in terms of HIV. The CRISPR-Cas system approaches by removing or inactivating the integrated provirus DNA from the host genome. The HIV-Rev encoding gene, or protein that's responsible for protein expression, is targeted by the CRISPR-Cas9 system, and the CRISPR RNA targets this gene to induce strong HIV suppression in infected T cells. CRISPR-Cas-based gene therapy can also block viral infection of already infected cells, also known as superinfection. HIV encompasses a mechanism to prevent superinfection by superinfection resistance. This mechanism is achieved through HIV-induced downregulating of the viral entry receptor with the viral entry receptor CD4. The viral proteins of HIV remain intact with the receptors in order to regulate the expression of CD4. CRISPR-Cas9 targets the first exon of the viral protein and uses 3 CRISPR RNAs to target the viral proteins by inactivating all infectious proviruses in T cells.
CRISPR-Cas9 holds the potential to bring about revolutionary advances in medicine, particularly addressing diseases such as AIDS and HIV. The CRISPR-Cas9 system is a versatile tool, but with it comes many ethical concerns. The advanced technology allows researchers to precisely edit the gene material of an organism, and this is very unnatural and presents a gateway to many ethical debates. Ethical concerns around the use of CRISPR technology are largely based on the ability to enhance human traits and make them almost like mutations that are really unnatural and therefore could be harmful to the person. CRISPR-Cas9 acts in a way that no other system can, and is able to edit the gene and the HIV genome within the infected cell. It's a major global health problem, and with CRISPR-Cas9 there can be a solution to this. So it is a preventable disease, and the benefits outweigh the severe risks associated with it.
Is this technology ethical? Like Sophie said, a large concern with CRISPR technology is the potential ability of enhancing human traits, specifically things that are not dangerous but are more for aesthetic purposes. However, when it concerns the application of it within AIDS and HIV, we believe the short answer is yes, it is ethical, especially in life-threatening cases when there are few options to choose from. CRISPR is an easy choice to make because it is there to affect the genes that are encoded for HIV and AIDS. And when it comes to the application of CRISPR and HIV and AIDS, the only potential for it being risk, or some people would think like ethical reasons, is the potential for accidental mutations that result in negative side effects, but the benefits do outweigh the risk.
Lastly, the conclusion. This was made by Carissa, but she's unable to film right now due to having a 5-hour volleyball practice. So in conclusion, CRISPR derived from the immune systems of bacteria and archaea stands as a groundbreaking gene editing technology that has been harnessed for disease prevention. The focused application on the HIV Rev encoding gene showcases its potential in inducing suppression of HIV in infected T cells. Despite the promising benefits, the field of gene editing is not without its challenges. Nevertheless, the potential to prevent and treat diseases through CRISPR technology underscores its significance in the ongoing discourse on genetic engineering, emphasizing responsible and ethical utilization of this powerful tool.